The Part 3 notes will be on the Calvin cycle. In the Calvin cycle, carbon will enter in the form of carbon dioxide, which is an inorganic molecule, and it's going to leave in the form of an organic molecule, a sugar, for example, glucose, C6H12O6, according to the equation. In this process, both ATP and NADPH that are made in the light reactions will be consumed. The carbohydrate that we will actually produce in the Calvin cycle is a 3-carbon sugar called triose phosphate, or TP, it can be abbreviated. Note that this is the same sugar that we talked about in cellular respiration. It was one of the intermediate molecules of glycolysis. In order to make a molecule of triose phosphate, the Calvin cycle will occur 3 times. Each spin on the Calvin cycle is considered to consume 1 CO2. That means it will take 3 spins on the Calvin cycle in order to make a triose phosphate. The Calvin cycle can be divided into 3 phases. Phase 1, carbon fixation. Phase 2 is a reduction phase, and Phase 3 is the regeneration of ribulose bisphosphate, which is our CO2 acceptor. In Phase 1, or carbon fixation phase, we're going to fix inorganic carbon dioxide to an organic molecule. That is what fixation means. Take something that's inorganic and attach it to an organic molecule. So we have inorganic carbon dioxide. It's going to be attached to this organic molecule, which is made up of 5 carbons. RUBP, or ribulose bisphosphate, is a 5-carbon sugar. So we're going to combine carbon dioxide with a 5-carbon sugar using an enzyme called Rubisco, and we are going to make molecules that are unstable. So that's going to be shown here. So carbon dioxide is taken in by the plant stomata, attached to our 5-carbon sugar, RUBP, and we will first form this 6-carbon molecule using the enzyme Rubisco. This molecule is very unstable, and it will immediately split into 2 3-carbon molecules. These 2 3-carbon molecules are glycerate 3-phosphates, or GP. So if we were tracking all 6 carbon dioxides from the atmosphere, then we would have 6 RUBPs using 6 Rubisco enzymes, creating 6 unstable molecules, and that would give us 12 of our GP molecules. Fun fact about Rubisco, it is actually the most abundant enzyme on Earth, so it's really important to our life processes because it allows for the plants to make sugars in the Calvin cycle. And remember that the Calvin cycle, once the plants have the sugar, then that sugar can be passed through the food chain to all other organisms, and this is how all other organisms get the energy that ultimately came from the sun. And it's found in really high concentrations in the stroma, since the Calvin cycle happens in the stroma, which is important because it actually works pretty slowly. It's not a super effective enzyme, especially when carbon dioxide concentrations are low. And so we're going to talk about this in more detail in the Part 4 notes, like why would CO2 concentration be low? And what would that do to the plant? So keeping the concentrations of it relatively high are important because it would speed up that process when CO2 is low. In the next phase, phase 2, the reduction phase, each GP, or glycerate 3-phosphate, and you do need to know the full name of that molecule, glycerate 3-phosphate, receives an additional phosphate from ATP, and that's going to form an intermediate molecule. You do not need to know the name of this molecule. You just need to know that there is an intermediate molecule formed here because ATP is spent. So here's our glycerate 3-phosphate, or GP. It's our 3-carbon sugar that we made in the last phase. ATP gets dephosphorylated, and we phosphorylate our GP molecules. After that comes the reduction. NADPHs will be oxidized in order to reduce this 3-carbon molecule. Once this 3-carbon molecule gets reduced, we've now made triose-phosphate, or TP. Remember that triose-phosphate is our end product of the Calvin cycle. So are we done with the Calvin cycle? Nope, we still have another phase. Okay, so since, like I said before, it would take 3 carbon dioxides in order to make a 3-carbon sugar as our final output, so 1 molecule of triose phosphate, you typically see the Calvin cycle tracking 3 CO2s at a time. It will always be a multiple of 3. So it could be 3, could be 6, it could be any multiple of 3, usually 3 or 6, but typically 3. So if we were tracking 3 CO2s coming in, so that's 3 spins on the Calvin cycle, then those 3 CO2s would use 3 different Rubisco enzymes combine with our 3 molecules of RUBP, and you do need to know the name of this ribulose bisphosphate, combine with RUBP, that's 5 carbons in that sugar, and we're going to make a 6-carbon molecule, 3 of them, because we're tracking 3 of each of those coming in, 3 spins on the Calvin cycle. So 3 unstable 6-carbon molecules. They are not shown here because they are so unstable that they do immediately split into 6, since we had 3 of those, 6 molecules, 3-carbon molecules of GP, or glycerate 3-phosphate. Keep in mind that all these terms have lots of different abbreviations and even slight rearrangements of what they can be called, but I'm teaching you the IB terms. So it's hard to find a diagram that matches perfectly with exactly what IB wants, which is why we'll draw it on the board later. But I am just trying to reiterate them when I say it out loud. So again, 6 molecules of glycerate 3-phosphate, or GP, have been formed at the end of fixation. In the reduction phase, shown here, 6 molecules of ATP will be spent because we have 6 of these molecules that need to be phosphorylated. And so that's going to give us 6 of our intermediate molecules that now have an additional phosphate. These 6 molecules will get reduced into 6 molecules of triose phosphate. It will take 6 NADPHs to achieve that. In this step, we will also lose an inorganic phosphate. So 6 NADPHs will be oxidized. You can see them leaving here. And that's going to reduce all 6 of these. An inorganic phosphate also pops off, and we now end with triose phosphate. And we have 6 of them. This is the end product of the Calvin cycle. However, not all of them get to leave as the end product of the Calvin cycle. Of our triose phosphates now, you can see that, that one of them leaves the Calvin cycle as triose phosphate. This can be incorporated into a glucose or other organic compounds. And the other 5 stay in the cycle. They go on to the next phase. In the next phase, it's going to be the regeneration of our UBP phase. In this phase, these 5 molecules of TP get rearranged into 3 molecules of ribulose biphosphate. We're going to spend 3 more ATP here. This is the reason why it's called a cycle, because we start with ribulose biphosphate and we end with the same molecule. We remake it in the cycle. Let's count our carbons, make sure we have them all. So at the beginning of the process, we started with 18 carbons because we had 3 CO2s, so that's 3 carbons. And then we had 5 ribulose bisphosphates. So 3 times 5, that's 15. So here's my RUBPs, here's my CO2s. That's all 18 carbons. In the process, we're going to make 6 molecules of triose phosphate. Remember, triose phosphate is a 3-carbon molecule, so 6 times 3 is 18. Of those 6 triose phosphates, 1 leaves the cycle as the final product. The other 5 stay in the cycle. So we still have 18 carbons. Here's just another picture of it, showing all the same things. 3 CO2, so 3 spins on the Calvin cycle. They come in, they combine with 3 molecules of ribulose bisphosphate using the enzyme Rubisco. We're going to make, and what's not shown here again is we're going to make 3 6-carbon molecules that are unstable. They immediately split into 6 molecules of glycerate 3-phosphate, or GP. Each GP then receives an additional phosphate by spending 6 ATP to phosphorylate all 6. We're going to end with 6 intermediate molecules here. All 6 of those molecules will receive a pair of electrons from NADPH, getting reduced into triose phosphate, 6 of them. An inorganic phosphate will leave in this step as well. Of our 6 triose phosphates, 1 will leave the cycle. The other 5 must stay in the cycle and be regenerated into 3 molecules of RUBP again. It will take 3 more ATP in order to achieve this. I don't know what I did. Sorry about that. Click something. Okay, so this just shows it a different way. But again, 3 carbon dioxides enter. We will form our 3 unstable molecules that are 6 carbons in length. They immediately split into 6 molecules that are 3 carbons in length, called GP. These GPs will be phosphorylated. That intermediate is not shown. We'll make 6 of them. Each of those intermediates will also receive a pair of electrons and get reduced by NADPHs. It will take 6 of those, and therefore we will be left with 6 molecules of triose phosphate. 1 can leave the cycle and be incorporated into a glucose or other organic compounds. 5 must be recycled in the cycle. It will take 3 more ATP to do that, and we will then make 3 5-carbon sugars of RUBP, completing the cycle. Here's where the words of regeneration phase come up, even though we already talked about it. But again, our 5 molecules of triose phosphate that stay in the cycle go through a series of enzymatic reactions to create 3 molecules of RUBP, and this will require 3 more ATP to be used. This is one of the reasons why we need cyclic electron flow. Remember that the Calvin cycle is consuming a total of 9 ATP and 6 NADPHs per 3 spins on the Calvin cycle. That means we have to be able to make more ATP than we do NADPHs, right? So we have to be able to have this additional 3 ATP. Remember that in cyclic electron flow, we get that. We get ATP made, but we're not making any NADPHs. And so this allows for us to make that excess ATP that is required to complete the Calvin cycle. This leads to negative feedback, the slowing down of the production of NADPH so that we can continue to make enough ATP for the Calvin cycle. So we're slowing down non-cyclic electron flow and this negative feedback. Sorry, I keep clicking on websites. Okay, so again, we're consuming 9 ATP, 6 here, 3 here, and only 6 NADPHs per 3 spins on the Calvin cycle. This is showing the same thing. More ATP consumed because it's consumed in 2 parts of the reaction, 2 different phases on the Calvin cycle compared to NADPHs. So remember, the net equation for photosynthesis is 6 carbon dioxides plus 6 waters and light energy gives us C6H12O6 and 6 oxygen molecules. And now we know that we've tracked all of these, right? 6 waters and light energy are used in the light reactions, and that's where we'll see the release of oxygen. because of the energy molecules made in the light reactions, the Calvin cycle will use those along with our 6 CO2s, and they will be converted into a sugar. So what can the plant do with the triose that they make in the Calvin cycle? Something that's important to note is that we've been focusing on sugars made, which is good. They do need to make sugars and use those sugars, but something that's interesting is that all the carbon compounds that a plant is going to need, all those carbon compounds are made by the photosynthetic fixation in the Calvin cycle. So being able to fix carbon dioxide to our UBP and lead to a sugar, really important. So during fixation, most of the molecules will ultimately be used to produce the sugar. So we'll always make the triose phosphates, but then those triose phosphates will be used to make glucose. Glucose is the major energy for cells, so about half of those triose phosphates will in fact be used to make glucose. But glucose itself can be converted into a lot of diverse compounds. We can combine it to make a disaccharide of sucrose. Sucrose is important because it's used for transport of sugars. Remember, we have to make the sugars in the leaves, but then we have to get those sugars to all parts of the plant. So even the roots, for example, are going to need those sugars, and they don't photosynthesize. So sucrose makes for a good way to transport the glucose down because it's a disaccharide. It's going to be used less by the cells along the way. And we'll talk about that in more detail in the plants unit. Starch, we talked about starch in BioH, right? Starch is a bunch of alpha glucoses linked together, and they're going to use that to store sugars. They can store it in their roots, they can store it in the fruits, they store it in tubers, which are like potatoes. The seeds need to be able to store starch so that they have a little food reserve when they start to germinate. And then they can use beta glucoses to build their cell walls, right? Because they're going to link them together to make long chains of cellulose, which is a very strong, structurally supportive polysaccharide, which we talked about also in BioH. However, they can use those triose phosphates to do other things as well. So they can use the triose phosphates to make fatty acid chains and glycerol, which glycerol, remember, is a 3-carbon alcohol that we talked about in BioH, and they can link together glycerol and 3 fatty acid chains in enzymatic reactions to make fats or triglycerides. They can also, through enzymatic reactions, convert the triose phosphates into amino acids with the addition of mineral uptake. So they're going to need to be able to take in minerals from the roots that are needed for amino acids, because remember, amino acids are going to have nitrogen in them as well as sulfur sometimes, and so those would need to be able to come in to be able to be used in this rearrangement. And then remember, the other product of photosynthesis was oxygen. And so some of that oxygen in a plant can actually be directly consumed for them in cellular respiration, because they're going to need that as an input to cell resp. But any excess oxygen will leave through the stoma by diffusion. So it's just going to go out through the stoma, and this is the oxygen that we get to breathe in. And that is it for the part 3 notes.